Oh, okay. Cool. All right, so assalamu alaikum everyone. My name is Saleha and I'm a intern. Every time I have to introduce myself, I almost say that I'm a fifth year, but I'm an intern. Uh, and those of you who already know me, I taught in pre-med this year. So if you went in pre-med, you would, remember me, I hope. Anyway, so today I'm going to be teaching two lectures. The first is Electrophysiology of the heart, and the second one is Excitation, Contraction, Coupling. I know that these, when you guys taught them, they may have seen this, when you guys learned them first, they may have seemed really tough, but I promise you these lectures are pretty easy. And I made sure to pick out the most important things. I covered everything, but I made sure to highlight, some really important things that you guys would need to know for your exam, inshallah. Anyway, let's get started. Both lectures are we should finish in less than 2 hours, inshallah. I don't expect to take a full hour for each lecture. Anyway, one more thing. I have a tendency to speak really fast. So if I do start speaking fast, please let me know. Just be like, Saleha, you're going way too fast. And I would slow down. Okay. All right. So let me turn on my pointer before I start. How do you turn it on? Okay, there we go. First, we're going to talk about blood flow through the heart. I wanted to start off like in a very concise and specific manner. And hold on, just give me 1 second. I think my Wi-Fi is unstable, so I'll connect to a different Wi-Fi. 1 minute. Perfect. So starting with the objectives of this lecture, first we're going to talk about blood flow through the heart. Then we're going to talk about the conducting cells, the contractile cells, and autorhythmicity. Then we're going to move on to the cardiac conduction system. And we're going to discuss two really important things in this lecture. The first is the pacemaker action potential, and then we're going to discuss ventricular action potential. And these are maybe the two most important things in this lecture. Then we're going to move on to the absolute and refractory periods, and that's pretty much it. Okay. So starting with blood flow through the heart, just a quick recap, just to refresh your memory. Blood starts flowing from the atria, and the atria contract, blood goes to the ventricles. When the ventricles contract, blood goes to either the lungs or the rest of the body, right? So blood from the right ventricle goes to the pulmonary artery and to the lungs. Then it circulates through the lungs, gets oxygenated, and then comes back through the left atrium into the left ventricle, and then goes out through the aorta, right? So atrial contraction is what causes blood flow to move to the ventricles. Ventricular contraction is what causes blood flow to the pulmonary arteries and the aorta, or to the rest of the body. Now, one really important thing to keep in mind here is that the fact that the atria are contracting together and the ventricles are contracting together is what really allows blood flow to be, consistent. Imagine if the atria and the ventricles were contracting each independently, not in any kind of rhythm. The blood wouldn't flow consistently and not systematically. And what would happen as a result is the heart would be a very ineffective pump. It would not be able to pump to the whole body if different parts of the heart were not contracting simultaneously and in collaboration. So that's a very important thing to keep in mind. That point is going to be addressed later. Okay, coming to conducting cells, contractile cells, and autorhythmicity. So how does the heart contract? What really happens in the contraction process of the heart? The heart has two main types of cells. The first type is the contractile cells, or the conducting cells, or pacemaker cells. The second, also, I have a question. Are you guys seeing balloons on my camera? Because that keeps showing up for me. I don't know why. Every time I do a thumbs up, like I see balloons. Anyways, ignore that. I don't know why that's happening. Okay, so there's two types of cells, conducting cells, which are also known as pacemaker cells. And these are of two types. The first is the SA node, which is the green thing here, and the second is the AV node. They're both the same, but they're just at different locations at the heart, and they serve different purposes. These cells generate and produce electrical impulses on their own and cause a slow action potential. When I say that they produce electrical impulses on their own, this is what causes the heart to have its own rhythm, essentially. Because these cells, these conducting cells, are capable of exciting themselves on their own, they don't need any innervation from the brain or the peripheral nervous system or anything else. They're simply able to contract on their own, independent of the brain and the central nervous system. This is an important point, which I'm going to elaborate further on the next few slides as well. So that was the first type of cell, the SA and AV nodes, right? The second type are the myocytes, or the contractile cells. These cells contract after they receive the electrical impulse from who? The SA and AV node cells. So the SA AV nodes start the impulse goes down to the myocardial cells. The myocardial cells are the ones that contract in a systematic manner to then, cause the muscles to shrink and, essentially push the blood out of the heart. And when you compare the action potential of the conducting cells or the pacemaker cells to the action potential of the contractile cells, the SAAV node have a slower action potential, whereas the contractile cells have a much faster action potential. Okay. Okay, there's my laser pointer. All right. So I mentioned the conducting cells are capable of something called self-excitation. This is why they're known as the pacemaker cells, because they set the pace of the heart. They set the heart rate. So what happens is these cells are able to generate their own impulse. This impulse is then transmitted down the cardiac conduction system here, like this. And these specialized contractile cells, which are the muscle cells, they then contract to cause, a depolarization of the heart. Sorry, they depolarize to cause contraction of the heart. Okay. So this electrical system that I'm showing you here is independent of neuronal activity. The heart does not need external innervation from the nervous system to cause contraction. This is known as autorhythmicity. Auto means on its own. And rhythmicity is the ability to have a rhythm. So the heart rate or the heart rhythm is set by the SA node. Impulse goes from SA node down here to the AV node, and then it spreads further. We're going to look at each of these things individually in a systematic manner now. So now coming to the cardiac conduction system. The important components of the cardiac conduction system, someone asked a question, are conducting cells or non-contractile cells? Yes, conducting cells only transmit the impulse. They don't contract to push blood out of the heart. Contractile cells are muscle cells, and those muscle cells cause contraction. When they all contract together, they cause contraction of the heart muscle to push blood, let's say from the atria to the ventricles, or from the ventricles to the rest of the body. I hope that answers your question. Okay, so important components of the cardiac conduction system, starting with the SA node or the sinoatrial node. It's positioned at the top of the right atrium. Next, we have the Bachmann's bundle, which is really just an extension of the SA node into the atria, the left atrium. Then we have the atrioventricular node. So the impulses from the SA node directly go to the atrioventricular node. Then we have the bundle of His. So this is the bundle of His, which is technically an extension of the AV node, actually. And then after that, we have the Purkinje fibers, which are located in the ventricular muscle, essentially. So starting with the sinoatrial node first. The sinoatrial node is located in the right atrium, close to the opening of the superior vena cava. It functions as the heart's intrinsic pacemaker because it regulates the heart rate. The heart rate is set by the sinoatrial node. The sinoatrial node then transmits impulses to the right atrium and the left atrium, and it transmits relatively fast at a speed of 1 meter per second. Okay. All right. So the SA node transmits impulses to the atria relatively fast at a speed of 1 meter per second. Now coming to the atrioventricular node. It's located in the atrioventricular septum. It receives impulses from the sinoatrial node. They go down the internodal branches. These are the internodal branches, and they go to the AV node. The AV node, however, has a much slower rate of conduction. Remember how the SA node was 1 meter per second? Here it's 0.05 meters per second, so it's much slower. And what this means is it causes a delay in the conduction by about 100 milliseconds. And these 100 milliseconds are crucial because they allow time for the ventricles to fill up with blood before they contract. So the atria contract, blood is now going into the ventricles. But before the ventricles can contract, they need to be sufficiently filled up with blood, right? So this minor, teeny tiny delay, which we might think is insignificant, is actually very critical because it is what allows the ventricles to fill up with blood before they contract. All right, now coming to the bundle of His, it's an extension of the AV node. It receives impulses from the AV node and transmits them down to the ventricles. It divides later into the left branches, left bundle branches, and the right bundle branches. And these are the Purkinje fibers, which are at the very end. Each terminates as a Purkinje fiber. And they disperse, the Purkinje fibers disperse the impulse throughout the ventricles, ultimately causing contraction. So that's it for a summary of the cardiac conduction system. Now, here's a little, game thingy that I just made. Let's order the cardiac conduction system from start to finish. So I want you guys to type it in the chat for those of you who are listening, and I'm going to start going through them one by one. So the first thing, where does the cardiac conduction system initiate, or where does it start? Where does the impulse come from? It starts from the SA node. The SA node is where it begins. Perfect. Then it goes where? Remember I mentioned this briefly. It goes to the internodal pathway, which is linking the SA node and the AV node. So SA node, internodal pathway, AV node. Next, we have what? We have the bundle of His. That's the fourth thing, or the fourth in line. From the bundle of His, where does it go? It goes to the left and right bundle branches. Yep. And finally, it terminates as the Purkinje fibers. I think you guys all got it right, whoever is answering. Yep. Perfect. Okay. Yeah, wonderful. Okay. So this is a little GIF that I found, which kind of shows you how the impulse is dispersed throughout the heart. I think it gives you a really good idea of what it looks like. So that's really just it. Okay, now let's move to pacemaker action potential. We were done with half the lecture already. We're now coming to the most important thing. This one and the next thing are the most important, and then we'll be done. Okay, so this is a skeletal muscle action potential. Okay. This is not a cardiac muscle action potential, but I just wanted to throw it in there to kind of remind you guys of what an action potential looks like, because now we're going to be addressing a different kind of action potential. So this is just a refresher, if you will. So first of all, what is an action potential? In skeletal muscles, an action potential is what initiates contraction. It is a series of changes in the voltage of the cell membrane. And these changes are because of the voltage-gated ion channels that are present on the cell membrane. And every time ions go in and out, that generates a change in the cell membrane, in the membrane potential, which then causes the impulse to form. So in this case, these action potentials need an impulse from a neurotransmitter that is released from a nerve ending. It has to come from somewhere. They don't just happen on their own. But we know that in the heart, action potentials occur on their own. So there must be something different, right? Just keep that thought in your head. So just a further recap, the resting membrane potential is a voltage difference across the cell membrane in the resting phase. There is a threshold potential which has to be reached before an action potential can develop, right? So first there's depolarization, then there's repolarization, and then finally you have a little bit of hyperpolarization before it ultimately reaches resting membrane potential again. And once again, it is waiting for the next impulse from the neuron to, cause another action potential. Okay, Now coming to cardiac pacemaker action potential. This is what it looks like. Immediately you can tell it's very different, but it's not that different. It has some similarities in reality. So first, where is the cardiac pacemaker action potential actually occurring? It occurs in the SA node and the AV node, and it has only 3 phases: phase 0, phase 3, and phase 4. It has no stable phase. That's the main difference you're going to notice between the action potential, which had, a resting phase, versus this, which really doesn't have, it doesn't, there's no plateau or resting phase where it's flat. It's always going up and down, always flips. Okay, now coming to the most important point on this slide, self-excitation. How is self-excitation possible? Self-excitation is possible because these cells, their resting potential continuously moves upwards. There is never a time where it's steady. It's always, slowly repolarizing. And eventually when it crosses threshold, that causes an impulse or an action potential to form on its own. Yep. So this causes an action potential to fire. And this is why these cells are known as pacemaker cells. Okay, this is a slide from your professor's lecture. I just threw it in to highlight one thing, which is that the membrane potential is determined by the permeability of the cell membrane to different ions. That's all you need to know from this slide. What you need to know is the changes in the action potential happen because different ions are going in and out and in and out. And each ion has a different equilibrium potential of its own, right? So if the cell membrane is permeable to sodium ions and potassium ions and calcium ions at different points during the action potential, that is what determines really how the action potential shape looks like, which ion is coming in and which ion is going out. And it also determines the self-excitability of this because, for example, in case of cardiac action potential, sodium ions are continuously coming in towards the end, and then that eventually changes to calcium ions. And what we're going to notice later is that is what causes this continuous little slope upwards. And all of this is based on what? The fact that different ions are going in and out, and at any one point, the membrane potential of the cell is determined by which ions are crossing it. Okay, that's what you need to know from here. Okay, let's go through the different phases of the cardiac pacemaker action potential. So while it doesn't have a resting membrane potential, nothing like that, I am going to be referring to a resting membrane potential, but it's just like a figurative resting membrane potential, okay, like an imaginary one. So once the potential of the cell reaches above -55, so I would say it's about here, what happens is the depolarization phase starts, right? Or phase 0 starts. What happens is there is an influx of calcium ions, and this occurs through L-type calcium channels, which are slower than the sodium channels. The sodium channels close at -55 and are not involved in depolarization. Why am I mentioning sodium channels here? I'll get to that in a minute. There are sodium channels that are going to be opened here, but they eventually close, and then calcium channels continue. Once the threshold is reached, that is when the L-type calcium channels open. Here we have a different type of calcium channel. We'll see that towards the end. But here, when threshold is reached, a different type of calcium channel opens, which is the L-type calcium channel. These are slower than the sodium channels, and they open at -40, and they cause depolarization by allowing calcium to enter the cell. An influx of calcium ions, more positive ions are coming into the cell. That's going to raise the membrane potential from -40 to a more positive potential, right? Okay, so that's phase 0. Calcium ions are coming in through L-type calcium channels. Now let's move to phase 3. In phase 3, we have repolarization. So it's starting to become negative again. And this is because of an efflux of potassium ions. Efflux means potassium is leaving the cell. Influx means coming in. Efflux means going out. So here we have an efflux of potassium ions, and this occurs through delayed rectifier potassium channels. In your lecture given by your doctor, this is how he refers to them, IKR and IKS. They have 2 components. The those aren't really as important for you to know. What's important for you to know here is that it occurs through delayed rectifier potassium channels. Now the calcium channels have closed. No more calcium ions are coming into the cell. It's only potassium ions that are leaving. So think of it this way. Positively charged ions are leaving the cell. What's going to happen? The membrane potential is going to go from more positive. It's going to start becoming negative if more positive ions are leaving, right? So the cell membrane potential starts becoming negative. This is known as repolarization. And the inside of the cell is now negative. It's reaching the equilibrium potential for potassium. And there's no resting phase. So immediately we move on to phase 4. Phase 4. Now, this is the tricky part that I was mentioning earlier. It's a little bit confusing, so just stay with me. Phase 4 is a slow depolarization phase, and different ion channels are involved here. So the first type is funny channels, also known as IF channels, and they allow an influx of sodium. Okay, so sodium ions are coming into the cell through these funny channels. These are activated by two things. Hyperpolarization. What's hyperpolarization? When the cell membrane becomes really negative. So this is what activates these sodium channels, these funny current channels. And they're also activated by cyclic AMP. When cyclic AMP is high in the cell, it causes faster opening of these funny channels. And then the phase 4 starts occurring quicker, okay? So this is a slow, spontaneous depolarization. Phase 4 continued towards the end of phase 4 is when you have is when the sodium channels close, actually, and you have a different type of channel open. These are the calcium channels, and this time they're T-type calcium channels. They open at negative 55 millivolts. Now, when the threshold reaches negative 40, which is this line that you see here, when the threshold reaches negative 40, these T-type calcium channels are going to close. And what's now going to cause phase 0 is the L-type calcium channels, which are now going to open. So everything is closing and opening at a specific point. Let me just quickly recap. So sodium channels, funny channels, are open in phase 4. By the time the membrane potential is around negative 55, the sodium channels close. Now you have these T-type calcium channels that are open. T-type calcium channels close at negative 40, and then you have L-type calcium channels, which are open. These close when the cell membrane is, relatively positive. Then you have the potassium channels, which open, which allow an efflux of potassium. And finally, your sodium channels open again at the end of phase 3, beginning of phase 4. And this is what generates the next action potential. So you notice how, because there's always some channel opening or closing, something is always happening, there's always hyperpolarization happening. This enables the next action potential to occur on its own. The cardiac muscle cells, the cardiac pacemaker cells do not require any innervation. Now here's an interesting aspect. So whenever there's sympathetic stimulation, for example, when there's an adrenaline rush, what happens? How come your heart rate increases? Because your heart rate is being determined by the SA node, correct? So what happens when your heart rate increases? The phase 4 becomes shorter. It becomes shorter, and it's, like a steeper slope. And that allows the next action potential to be generated faster. So there's more action potentials. The heart rate is now quicker because it's being determined by the SA node. So the SA node is the fastest to depolarize. What this means is it sends this electrical impulse that is now being generated at the SA node is sent all throughout the heart, right? And this allows the SA node to be like the boss of everyone else, because the other cells, although they're also part of the conduction pathway and they're able to, have the same kind of action potential, their action potentials would be slower than the SA node. The SA node is the fastest to depolarize, like we said, right? So it sends the primary impulse before any other part of the conduction pathway can reach threshold. Therefore, it sets the threshold for everybody else down the line. This is known as overdrive suppression, and it's really important in, number 1, setting the heart rate, and number 2, preventing contractions from occurring on top of each other, okay? Preventing an arrhythmia, essentially, that's what this does. Okay, so a quick recap. If you guys want to answer along with me, that would be great. I'm going to show you an image and we're going to go through the different phases of the action potential. So there's 3 phases, phase 0, phase 3, and phase 4. Which ions are responsible for each phase? Let's start with phase 0. Which ion is coming in or going out, and what type of channel are we talking about in phase 0? Here, we're looking at depolarization, right? And what's happening is there's an influx of calcium ions through L-type calcium channels, right? Yep, perfect. Okay. Next, we have phase 3. I'm going to wait for someone to type the answer before I say it. I'm not going to give it away this time. Phase 3 is, what is it and what ion are we talking about and what's happening? Okay, so phase 3 is repolarization. And it is through, yes, it is through the potassium, it occurs through efflux of potassium ions through delayed rectifier potassium channels. Perfect. Okay, next we have phase 4. Phase 4 is the spontaneous depolarization, which is responsible for the self-excitation of the SA node. This happens through an influx of sodium channels through specialized spiny channels and some influx of calcium at the end through T-type calcium channels. Okay, cool. Now let's move on to ventricular action potential. We're at a good pace. If you guys have any questions at any point, just mention them in the chat and I will try my best to answer them as we go, okay? All right. So coming to the cardiomyocyte action potential or the contractile action potential. Okay, so here we're looking at the action potential of the ventricular myocyte, and this occurs in the myocardium in the bundle of His and the Purkinje, not the Purkinje fibers, but essentially just the myocardium. It has 5 phases. Self-excitation is not possible. This needs an action potential from the pacemaker cells or the adjacent cardiomyocytes to allow the membrane potential to rise above -90. So here there is a resting phase. This is -90 where the resting membrane potential is. Whenever there's this, an impulse from the conduction pathway, this then causes these cells to contract. This causes a new action potential in these cells. Okay. Also, quick reminder, this is a correction. Ignore bundle of His and Purkinje fibers. It only occurs in the myocardium. Okay. So let's start with phase 0. What happens in phase 0? There is depolarization due to influx of sodium ions. And hold on. Yeah, there we go. Okay. This occurs through voltage-gated sodium channels. Now, this membrane potential goes from -90 all the way to +20. Another ion is also involved here. This is the calcium ion, which is involved at the end of phase 0, and it goes towards the end of phase 0. And again, we're looking at L-type calcium channels. The L-type calcium channels are back. Okay, so here we have an influx of calcium as well. Calcium is entering the cell towards the end of phase 0, and this helps the cell to reach +20. Okay. Next, we have phase 1. So phase 1 is called the little peak or the little tip of the action potential, also known as the initial repolarization. So what happens here? There is an efflux of potassium ions. Potassium is leaving the cell. This occurs through transient potassium channels called Ito channels. And in this, at this time, the Na+ channels or the sodium channels that we were looking at earlier, these ones are now closed. And because potassium is leaving, a positive charged ion is leaving the cell membrane potential starts to become more negative, right? Okay. Now keep in mind that calcium channels are still entering the cell over here. Because these are L-type channels, these are slow to open and they're also slow to close. Remember how we mentioned earlier that there are some channels that, essentially the membrane potential is determined by which channels are open when. So keep that in mind. These channels are still open at this point. So calcium is still coming into the cell. Potassium is leaving, and at the same time, calcium is coming in. That brings us to our next phase. This is the plateau phase. It is a flat phase, somewhat, and this really happens because potassium is leaving at the same time that calcium is still coming in. There's a little bit of calcium still coming in. So what happens is it reaches a kind of flat line. Phase 2 is a plateau. So calcium is still leaving through delayed rectifier potassium channels. And the calcium is coming in through the L-type calcium channels that are still open. And this results in a sustained depolarization phase. Now we come to phase 3. In phase 3, the calcium channels are now closed. They're no longer open. What that means is now the potassium has overpowered the calcium. It's leaving the cell still, and now this causes a continued repolarization. So there's persistent outflow of calcium, which exceeds there's persistent outflow of potassium, which exceeds the calcium that was coming in. Also, the calcium channels are now closed, so the repolarization continues. And it goes down all the way to resting membrane potential. So a quick recap of everything. Phase 0 was what? Sodium channels were opening, and these were voltage-gated sodium channels, and sodium was coming in. Next, towards the top, we had calcium channels opening. The L-type calcium channels, which are a little slow, and they help the resting or the membrane potential to reach positive 20. When it reaches the peak, this is where potassium ion channels open. Delayed rectifier potassium ion channels open, and there's an efflux of potassium from the cell. Now, phase 2 was what? It was a plateau phase where potassium is leaving. At the same time, calcium is coming in, so there's a sustained depolarization. Finally, we come to phase 3 where the potassium wins and only potassium is coming in. Calcium channels are closed, and there is a repolarization all the way to phase 4, which is the resting membrane potential. Okay. So this was a slide from your doctor's presentation, transmural differences in ventricular action potential duration. This was essentially about how the action potential is different in the endocardium versus the epicardium. What you need to know here is that the epicardium is the last to depolarize. It's also the first to repolarize because the impulse comes from the endocardium. So the endocardium depolarizes, then it spreads outwards to the epicardium. The epicardium depolarizes last. It also repolarizes first, and that goes back to repolarization, and then it spreads to the endocardium. And that's really all that you need to know from here. The purpose of this is because there is a greater expression of Ito channels in the epicardium. Now, if you let me go back a few slides, the Ito channels were the ones that open in the initial repolarization channels. So when there's more of these channels, the initial repolarization occurs quickly. And here you'll see that in this image here. So it is like more of a notch. Instead of a peak, you now have a notch. And that's what happens in the epicardium. And that's really it for that side. Now coming to the very last part of this lecture, the absolute and refractory periods. This was always something that I really enjoyed learning because it made a lot of sense to me. And I would always think about how subhanAllah, without this built into our systems, we may not be able to survive. It's crazy how Allah has really perfected us down to the tiniest things that you couldn't even think about. Okay, so coming to the refractory period. The refractory period is a time during which if a second stimulus were to come, it would not be able to elicit or initiate another action potential. So what does that mean? This is the refractory period. If a new impulse comes anytime during this refractory period, a new action potential cannot occur. So let's say the action potential, the existing one, is now here, okay? We're now here. Let's say another impulse came over here, okay, like this, at like 300 milliseconds. It would not be able to cause a new action potential to occur because it's still in its refractory period. Now, what is the purpose of a refractory period? It regulates the firing of these impulses. It prevents impulses from coming back because they can't overlap. So it prevents that. and it allows the ventricles to fill up completely before the next contraction occurs. So this allows the heart to really be effective at pumping blood to the rest of the body. What is the absolute refractory period? It is a time where no matter how strong of an impulse you get, it's about like from 200 milliseconds, let's say 350, 370 milliseconds. So this period is when no matter how strong of an impulse you get, no new action potential can be generated because the fast sodium channels are deactivated currently. Remember, they get deactivated here and they were open here, but now they're deactivated here. And so they're so close. No new sodium can enter the cell. Okay, so a strong stimulus, no matter how strong it is, cannot cause a new action potential during this period. Now, a relative refractory period, also known as RRP, is a time where a new AP may be generated if a really strong impulse is transmitted, because now, towards the end over here, towards the end, sodium channels are starting to open again, the fast sodium channels. And what this means is if a new impulse were to strike at, let's say, 450 milliseconds, it hasn't reached resting membrane potential yet. But let's say if that happened, this might, if it was a much stronger impulse than normal, this might actually cause a new action potential to occur. Okay, so it must be stronger than normal. That's a condition for the relative refractory period. Okay, and that's just the purpose of refractory periods again that I've just repeated. Coming back to cardiac contraction, so one last important thing is that all cardiac myocytes are connected to each other via gap junctions. These gap junctions are called intercalated discs. And what happens is these gap junctions, because all the cells are close to each other, they allow the impulse to spread from one cell to another uniformly, which then allows the atria to contract together and the ventricles to contract together, therefore allowing the heart to be an effective and efficient pump for the body. Okay, that's it for this lecture, but I have a few questions for you guys. The questions are easy, but I should mention that they are very important. It's very likely that similar questions would come in your TVLs or your exams. Okay, so the essay note is A, group of conducting myocytes on the right atrium, or B, a group of myocytes at the superior vena cava / right atrium junction, or C, a group of specialized conducting cells at the inferior vena cava / right atrium junction, or D, a group of specialized conducting cells at the superior vena cava right atrium junction. Someone said, do we need to know about the effective refractory and supernormal refractory periods? Supernormal refractory period is not as important, but effective refractory period is essentially the large refractory period that I showed you in the beginning. I later split that up into absolute and relative, but that large refractory period is the effective refractory period. Okay, I see a couple of people answering. Someone said D, another person said D. Correct. D is the right answer. A sinoatrial node is a group of specialized conducting cells at the superior vena cava / right atrium junction. The next question. The sinoatrial node is called the pacemaker of the heart because of what? I'm going to let you guys read this. Is it A, that it allows both atria to contract rapidly in response to a signal from one node? Is it B, relays the regulated impulses through the intrinsic conduction system? Is it C, initiates the heartbeat through cells that can depolarize on their own? Or is it D, has unique muscle fibers that can function without external stimulation? Someone said C. I want more people to answer. Come on, guys. Come on. Another person said C. Okay. Let's go with that. C is correct. Well done, guys. Okay. This is the last question, I think. Yeah. Okay. Yes. So the delay of the AV node supports coordinated blood flow through the heart by what? Is it A, allowing the atria to contract simultaneously as soon as the SA node. Someone said D. Is it B, increasing the power of the impulse to maintain its flow through the bundle of his? Is it C, triggering the ventricles to expand in rhythm with the impulses from the bundle branches? Is it D, allowing the atria time to push blood into the ventricles right before the ventricles contract? So one person said D, but I want more answers. Another person said D. Someone else said it is D. Correct. Mashallah, you guys are doing really good. Inshallah, you guys do well in your exam. Okay, that's it for this lecture. This is my contact information, and these are the resources that I use. Also, I would highly recommend these videos. They were really, really helpful for me when I was studying for my step one, and also when I was making this lecture, I made sure to incorporate some of the photos from those videos because they're just so simple and so easy to understand. So I would definitely recommend those.